Thank you very much for a kind introduction, Dr. Willison. No one introduces speakers better than Dr. Willison, and I'm very honored to be here. I previously had the opportunity to speak to you about issues like the molecular albite and issues about unloading the failing heart from metabolic stress. And today I will take a little broader view, and I will share with you some of my personal experiences Dr. Willison already referred to. As being a graduate student, the last graduate student of Professor Hans Krebs at Oxford, and how this influenced my thinking about the heart and about metabolism in general. And my aim is to, because metabolism isn't that popular actually amongst cardiologists, my aim is to make all of us in the next 45 minutes experts in metabolism. When we come to think of it, our heart is the size of contracts 100,000 times in the course of a day. A lot of energy, and of course, you know, the energy. But the stores of ATP, the amount of ATP makes and uses in the course of the day, in the course of the day. That is cardiovascular. This is, you know, all this in flux because the heart doesn't store its ATP. So, of course, the most striking example is ischemic heart disease and in the heart. Metabolism and contraction of the heart are inextricably linked. Problem is, this is not that easy, and it's challenging. But I like to get across and make all of us experts in the next 40 minutes now. But before I do this, I'd like to share with memory of one of the giants in the field of cardiovascular physiology and metabolism. That is Lionel Opie, whom I was told on the way before I left here, passed away at the age of 87, just a few days ago. The date was not given. It was a colleague at the NIH. Lionel was one of the pioneers in cardiovascular physiology, but especially in metabolism. He discovered a number of major metabolic pathways in the heart and became very involved in metabolic support of the failing post-ischemic heart and also on the metabolic consequences of beta. I remember my first meeting him when he was in South Africa, when I was a fellow at the Brigham, and he professor or a visiting scientist. His hair was shoulder long. He was a hippie, and he was in about apartheid system in South Africa. That was Lionel, and in any case, back to metabolism. You have seen from objects that I've passed around the flux of fuel in the heart is no different actually from the flux car engine. This is a car engine, which is a converter of chemical energy into mechanical energy. The difference between the heart and the car engine is that the heart is much more efficient, number one. Number two, you don't have to go out and buy a new heart every 10 years. It renews itself from within each cell. Cell takes down this and old proteins and replaces them with new and functional proteins in order to keep that whole machinery going. Words, chemical energy to mechanical energy. So we're replacing the car engine now with a real beating heart, and we already what I alluded to, hearts are inextricably linked. Simple, in the system of homeostasis, fuel and oxygen goes in, and CO2 and water comes out. And this is actually the way we measure metabolic fluxes. We label fuels or oxygen, and we measure the labeled CO2 and the labeled water that comes out. So a matter of checks and balances is we can actually establish the stoichiometry between energy going in and energy coming out. Called thermodynamics, energy can neither be made nor be destroyed. So this is a very tight and very rigorously tested system. Okay, now this system of homeostasis allows the heart to respond to a whole series of stresses. And these thermodynamic stresses that you deal with when you relieve the pressure of the left ventricle and aortic stenosis with the power. In any case, then the neurohumoral stresses, which we all effectively treat in our patients with beta-adrenergic blockade, ARBs, ACE inhibitors, and aldosterone antagonists. Inflammatory stresses that were emerging targets now for treatment of heart failure. There are metabolic stresses, as we have learned, and my previous talk here was on metabolic unloading of the heart, presaging the craze about SGLT2 inhibitors now. This was what I talked about here was 6 years ago, long before the SGLT2 inhibitors became the darling of the heart failure and diabetes community. Then we have, of course, ischemic stress, and there is something that can be done about that as well, and a lot. So, but besides of my animation. Okay, besides, let's do it down here. Okay, there. Okay. Yeah. Fine. Besides the converting energy, metabolism also has a number of extremely important functions, and they deal with signaling and with anabolism. That is building up of contractile proteins, building up of the membranes the cell needs to function properly, to keep the different compartments apart. And so metabolism is the source of the redox state, signaling, building blocks of proteins, of phospholipids, and metabolism provides the modifiers of protein, of RNA, of DNA, and as we know now, of chromatin as well. And those are the proteins that most of them are enzymes that catalyze, again, metabolic reactions, but are subject to post-translational modifications. So there is a plethora of activity going on in the heart muscle cell that we only begin to understand now. But the problem with metabolism is that, like in this cartoon here, all the investigators seem to have a favorite subject, a favorite performer. And metabolism is incredibly fragmented, number one. Reflected in some of our seminars here and nationally at the meetings and medical center. And it's reflected also in our thinking, because as clinical practicing cardiologists, we don't like to get into the depth of the complicated metabolic system, and it's very understandable. Second problem, the drama of metabolism, that is incredibly complex. And this year, this of the metabolic pathways is about 50 years old, and there have been new metabolic pathways being detected. We can, of course, turn off and say, this is far too much, and somebody will have to sort this out for me, but I can't handle a complex system like this. Or we can take a stab on taking out some principles and understanding some principles. And one of these principles of energy transfer is actually intermediary metabolism cycles, like the cycle. That is in contrast to the common understanding that we have, also the linear understanding. When we drive a car, we have wheels, of course, or cycles, but we measure the distances and we measure the speed, the distance we cover in a certain amount of time. But we don't intuitively think of cycles. And I think that was the contribution of a young physician scientist who has faced a number of obstacles in his career, and I will allude to them in a moment. And every time he hit a roadblock, he had a new idea, or had an idea to fit together what he observed in his experiments. At the same time, he was a medical resident in Freiburg, and later on a faculty, a junior faculty, was looking after patients during the day. And in his off time, he wasn't married yet, he had a lab, and he had a medical student first, and then 2 medical students, and then 3 medical students, eventually also a technician and his lab. And that was Krebs. So this is Krebs, the way I know him, when he was 80 years old, on a conference in Bermuda, where people rent, 2 cycles, the urea cycle in front and the citric acid cycle in the back. Very kindly looking, modest gentleman, but I'll tell you, as tough as nails. And I'd like to tell the story. He worked until, essentially until the day he died, but he had to be in the hospital for a few days, and 2 days before he passed away. I visited with him and he asked me, have any new results? And I said, no, there's hardly any time. I'm done with my thesis and had defended it, and no. And then he said, well, I think you should leave now. You're wasting my time. Okay, so how does a physician scientist from about 100 years ago go through life? Well, Krebs was born at the beginning of the last century. His father was a ENT doctor in a small city in northern Germany, and his mother was a homemaker. At the end of World War I, military. A little bit as a medical student, and after he graduated, he got a job as a postdoctoral fellow with Otto Warburg, and Otto Warburg was, well, essentially the person who invented biochemistry at the time. There were a few others, but Warburg was the central figure and still is. There are about 10 different labs over at MD Anderson because we discovered something very, very important in cancer cell metabolism that are working on the Warburg effect as I'm speaking. And we have a metabolism interest group at MD Anderson that's attended every 3 months by 80 people. An amazing interest in cancer cell metabolism. Warburg. But then Warburg told him that his performance, told Krebs that his performance was not really up to for a full-time laboratory career. He should go back to clinical medicine and complete his clinical training, and this is where Krebs then became independent. He was very disappointed. It was taken in or received a job. Siegfried Tannhäuser. Siegfried Tannhäuser later emigrated to Boston and became the chief of medicine at New England Floating Hospital, Tufts University. Then, while Krebs worked independently, he published an important paper. Based on his research, he was immediately taken onto the faculty and got full tenure. And that was the discovery of the urea cycle. Then something happened that I will allude to in a minute. And Krebs had to leave Freiburg and found a job in Hopkins lab at Cambridge. Garland Hopkins was the biochemist in Britain. There Krebs started his independent career in Sheffield. Covered there the citric acid cycle. Won the Nobel Prize in 1953. Oxford always goes safe. They only take Nobel Prize winners for the Oxford, and became the biochemistry. And after retirement, he continued very active in research, died, and then his last original paper, the paper for which he did the experiments at the bench, for which he had applied for grant support, for which he analyzed the data and wrote the paper, was published 2 years after his death. So you can imagine how they've been going as a dynamo like Dr. Willison. So how does the promising young physician scientist, medical student. Pictures from Krebs's biography. What I always find amazing at the time, this was the 1920s, what the respect people had for learning and reading. They dressed up for it, you see? They dressed up for it. This here is a picture much later, after World War II. Warburg and Krebs together at the meeting. Warburg and Krebs, they were not exactly friends, but Krebs always referred to Warburg as the one who provided him with a toolbox for his research and that. Now, Krebs did something very unusual, I think, and very smart. In spite of his engagement in bench work, still until he was over 80, he also set aside time to write a biography, a short biography on his teacher Warburg, and his own autobiography. And so he left shortly before he died a documentation of the events as he saw them and he lived. Biography, this is the one the people, I have a copy of it, the people over at MD Anderson, our colleagues, they're salivating about it. Scanned it and scanned it and scanned it. This is the one. What Krebs wrote in there, in the preface, I'll share with you because it is amazing. He wrote in the preface that the passage of time will, I believe, increase. Warburg had already won 2 Nobel Prizes because the contribution. Warburg was an eccentric, everybody. Also able to convince the Nazis that he would find the cure for cancer. And Hitler was afraid of dying from cancer. So Hitler declared Warburg, who was Jewish, not Jewish, and supported Warburg to the very end. Irritating personal qualities, which probably true. Somebody who refused to write letters of recommendation for anybody, who refused to travel or to give lectures unless it was absolutely necessary. This was what Warburg is famous for. Complicated, but it's nothing but about here, and this is what he took with him when he set up his lab in Freiburg. This is not a linear accelerator. This is simple glassware that led to some of the. This is another sort of personal sideline. This is Freiburg. This was after the economic crisis in the 1920s. The German government made an effort to rebuild scientific or research infrastructure. Hospital and laboratories, and the accommodation for the residents, which was, I think, quite comfortable at the time and allowed people to function as physician scientists. So the first original observation was a cyclic process, the urea cycle. a cyclic process that he thought of based on the experimentals he got with urea synthesis. Ornithine passes through a circuit corresponding to the following scheme. And this was drawing of a metabolic cycle that proved to be correct. In fact, the establishment, the biochemical establishment, so excited about it that editor of the biochemical journal, who discovered beta oxidation of long-chain fatty acids, wrote to Krebs a letter. I mean, this was a young, medical resident. I thank you very much for your beautiful paper. Well, we don't get letters like this nowadays. Then here, your paper arrived here on the 14th and will soon go into press. I'm very anxious to see what else you will bring forth and can only congratulate you heartily for your results up until now. Amazing, right? Why a young, here, one of our cardiology fellows, Stratus Parkus is here, his age, right? And this is a letter the young physician scientist got from Franz Knopf, and I show this picture. Covered the oxidation of long-chain fatty acids with tracer methods at the time, but he was a German, as you can see here, and when the Nazis came to power, he quit his job as chairman of biochemistry in Tübingen in solidarity with his Jewish colleagues. This is the lecture handed in his thesis for tenure. A month later, Hitler has come to power and dismissed all civil servants, all faculty, university employees who were of background in one way or another. It didn't have to be, practicing. Krebs was not by any. This was 1933. Hitler had already announced before 1928, science, if the dismissal of Jewish scientists means the annihilation of German science, then we shall do without science for a few years. That's what can happen anytime. A terrible thought, but also very stressful for the young budding physician scientists. Krebs always considered himself a physician first. But the consequences, of course, of Hitler's edict was a lot of scientists emigrating, time to emigrate to Britain, France, Britain, United States, Mexico. Actually wrote a book on that she called Hitler's Gift, because there would be no Manhattan Project without the exodus of the German physicists. And interestingly enough, they made 1,700 scientists went to Britain, 1,400 to the United States. Of those 1,700, 28 future Nobel Prize winners, and 14 of them in the UK. One of them was Heinrich Taegtmeyer. Others. Okay, now this is now Krebs' first independent position as the lecturer in pharmacology at the Northern England. Here is a second cycle was published, and that is the citric acid cycle, and that's from a metabolic perspective, energy transfer perspective, the more important one. That was in 1937. Allow us to outline the principal steps in the oxidation of sugar. Not so terribly long ago. Acid cycle, and this is what Krebs published in his autobiography. Submitted the concept to Nature, rejected his paper. Scholarly language, you can read it. 1988, that is 7 years after editor of Nature apologized publicly in Nature. Mistake in the scientific publishing history. Krebs turned it around, submitted it to an unknown journal, Enzymologia. Unknown then, it still exists, and is unknown now. This is a way, I think, to get around, disappointments and hardships, or just find another way through this, navigate through the system, navigate the system. That's what he was. And then, of course, I quite enjoy in my sign a copy of his Nobel lecture. And interestingly enough, in 1980, when Krebs others organized a symposium, a big symposium biochemistry, and they circulated among the participants major function of the Krebs cycle, and offered 5 different answers. And they asked Krebs as well. Krebs scribbled on a piece of paper. There are about 24 major functions. Expand on this a little bit in the next 5 minutes. Other functions. Otherwise, the question is okay. Not easy to read. I remember when we went through the drafts of my paper, he sometimes scolded me that I couldn't read his handwriting because he couldn't read it himself. So, if I'm forced to make a choice, I would pick number 2. Hydrogen atoms from the medium. Quantitatively, it's the most, actually based on existing dehydrogenation theory in organic chemistry. Won the Nobel Prize for the discovery of bile acids, and was one of Krebs's teachers. This is teachers, and this is what happens actually in this cycle. FADH and fed into the respiratory and the phosphorylation of ADP. Animated. Janet, who is faculty now at the Technikon in electrons in protons, and the protons are pumped out to the intermembrane space of the mitochondria, and then the proton, that is transfer of energy is tied. Happens at incredible speed in order to keep up. Where's Dr. Willison's book now at the very end? Or do you run away with it, please? It's the 6 kilogram Dr. Willison's textbook a day in our hearts. Amazing, isn't it? So, towards article on evolution of metabolic cycle. Optimal, and cycles are optimizing the resource. And if you look around a little bit as a cardiologist, you will find that there are not only the citric acid cycle, there's also the circulation, which is a cycle, right? And then inside the mitochondria occurs the ATP cycle, and then the cross-bridge cycle, all cycles. So there is quite a bit. And the circulation, this is something, an issue, when to transfuse and when not to transfuse, and big issues. Now, a few years ago, when the Winter Olympics were in Squaw Valley, California, it was a phenomenal finish. Who won 7 medals. Wilson and I were still young and watching the sports news. Now this man, he was doping. He had hemoglobin of 17 or 18. Only the time, roughly when he died, but truncated erythrocytosis honorated him monument. Right, there is, so filling up a cycle, that is the circulation, right? So now the other side of the coin, of course, is the infamous one that I will skip over now, who used the same principle, actually, by injecting. So we have then put the cycles to work in the isolated working right heart, and this is construction here. This is a lot of work for my PhD thesis. Glassware, making the glassware and designing, and then getting the heart working and working like it were inside the body. Just a working. A lot of other things as well. We were interested in not on the membranes, but in the matrix. Investigated the metabolism now again, and is a reflection of mitochondrial matrix metabolism. And to our surprise, we found kind, but picked up again when glucose. There is a break actually in the finish to a process that's called anapleausis. But what I'd like to show is, Raymond is one of my pride and joys from 30 years ago or so. And he's now a professor of cardiology at Brown University or so. But we were rolling at the time. We had 3 JCI papers, one after the other, to prove this process of anapleausis. Carboxylated. That process is called anapleausis. That leads us to a number of anabolic function, biosynthetic function of the Krebs cycle that we are working on today, a lot in my lab. And the message is really discovery. Discovery of that vitamin C, but this is irrelevant now, but he was also a bit of a contrary thinker. Discovery is seeing what thinking what no one thought before. And this is really the backbone of work. Now, lights, RNA function, DNA function, is regulate actually the gene expression pattern of the cell. So this now all of a sudden puts metabolism, that has always been viewed as the servant role to keep the cell functioning, actually also into the driver's seat. Now, the genes in gene transcription is, of course, sometimes grabbing into the steering wheel, but the metabolism is in the back. In this work now, Anja Karlstedt, a current postdoctoral fellow, K99 award winner on a new metabolic pathway in the heart, onkometabolism, onkometabolite, affects the flux of the Krebs cycle and makes the Krebs cycle go backwards, actually, and generates now signals that acetylate histones and also methylate the DNA. So it's a very exciting thing on that. Yes, and number one on circulation research, and so this has led us now to think of a lot of other metabolic cycles, including phosphorylation, dephosphorylation, and all the things that are going on inside the cell, and that are reflected so beautifully in our medicine, cardiovascular research seminar series every Thursday from 4 to 5. Everybody is invited to those. And we're drawing the Krebs cycle. One more word about another important cycle, discovered also by another famous refugee scientist, Rudolf Schönheimer. That's the dynamic state of body constituents, the cycling of proteins, protein synthesis and protein degradation that renews, self-renewal, intracellular self-renewal. Every cell in our body, and especially our heart. We have done radioisotope tracer studies. He was in the lab. And others have since then confirmed that, showing that if you tag a protein, it is degraded to the extent that the heart that is beating in Dr. Willison's chest today, and all our chests today, on the 21st of February, is totally different from the heart that has been beating in our chests on the 21st of January. Each protein has its individual half-life, and each lot of work that still waits to be discovered. So this is the amino acid cycling, and this is some of the work that we have done recently, and I don't have time to do more than acknowledge people in my lab who have contributed to some of the principles that I have referred to in my talk, and of course, be nothing without support from the NIH. And this year it will be 44 years that I've been fortunate to be or once also the American Heart Association and the including the McDonald Fund here helped us at one time. Okay, now this is what Krebs wrote across the draft of my thesis when I handed it to him. Main conclusion missing. So what's the take-home message? Number one, energy homeostasis, metabolic homeostasis is a defining feature of every living cell. No life without cycles, except for some naughty medical students who put this sign underneath here, a bicycle rack. It says here, cycles attached to these relics will be removed. And the second, of course, take-home message is common diseases like diabetes, heart failure, cancer, are systemic diseases of disrupted cycles of disrupted metabolic homeostasis. I thank you very much for your patience and for your interest.